Welcome back to the deep dive. You've brought us a really fascinating stack of sources today. Looks like we're pulling together threads from biological development, focusing, it seems, on that little nematode superstar, C. elegans,
Hmm.
and also how modeling and, well, even physics helps us understand it.
Yeah, it's quite a collection. We're looking at, the whole range. From the real nitty-gritty of tracking single cells, all the way to building these complex networks. And then there's the actual physical forces in tissues, the molecular stuff, and the computational tools you need for all of it.
Exactly. So the plan today, our mission, is to really dive into these sources, pull out those key insights, those aha moments,
Mm.
and try to connect the dots across these, well, seemingly different areas, give you a clearer picture, hopefully, without getting totally bogged down in the weeds.
Right. The idea is to make this, it's a complex field, make it a bit more accessible, and really highlight the key discoveries and approaches that are in your material here.
Okay, let's unpack this then. So when we talk about studying development at this level, how do researchers even start? What kind of data are they actually capturing about an organism like C. elegans?
Well, it really starts with observation, mainly through microscopy. The core data, like the sources describe, it's typically tracking individual cell identities, marking specific time points, and then recording their 3D spatial coordinates, so their XYZ position.
So literally just following cells around in 3D space.
Pretty much, yeah. And what's really powerful about C. elegans, why it's such a great model organism, is its remarkably deterministic cell lineage. It's quite predictable. From the very first divisions, you can basically predict which cells will pop up. An early cell, say AB, it always divides into ABP and ABA every time. So that makes identifying and tracking the who of each cell much more reliable, across different worms.
Got it. Okay. So you know the cell's name, its identity, and where it is over time. But even with that deterministic lineage, I imagine the actual position isn't like perfectly nailed down every single time. The sources mentioned variability.
That's exactly right. The 3D coordinates, they're relative to the microscope images, right? And
Yeah.
cells aren't just sitting still. They're shifting, moving around, migrating inside the embryo. So while the lineage itself is fixed, the precise spatial coordinates, yeah, they show variability from one image frame to the next or one worm to another.
Okay. So
Right.
how do they like handle that variability, that movement?
Right. So researchers use techniques to normalize the data. Often this involves centering the coordinates relative to the whole system, maybe the center of the embryo.
Right.
Or they might average positions over a few time points, use some fancy tracking algorithms. But getting that refined spatial and temporal info is absolutely crucial for building the models later on. Both, spatial graphs showing who's next to whom, and the lineage graphs, the family tree.
And is this kind of super detailed data actually available for other scientists to like download and use?
That's a key point in the sources, yeah. The push for open data. So data sets like the full body transcription factor expression atlas for the L1 larval C. elegans, they're out there, complete with resolved cell identities. And that's huge because it lets other groups build on that work.
What form does that data actually take? It starts as images, right? But then what?
Yeah, it starts with microscopy images. Then it gets processed into, say, point clouds showing cell positions, or used for point matching, which helps follow cells between images. Often, this info gets organized into tabular data. Think like a giant spreadsheet, basically. Columns for cell ID, time point, XYZ coordinates. Oh, and they also use something called bipartite graphs, specifically for that cell matching step between frames.
And you say it's open, like really open.
Yes, the sources point to places like Zenodo, open repositories, and even formats like Docker files or MATLAB files that bundle sample data, testing data, so you or anyone could potentially download the actual data and the tools used in the research.
How does this specific L1 larval data, how does it compare to maybe older stuff, like the famous Waterston data set?
Good question. The classic Waterston data focus heavily on the embryonic stages, with names like AB, EMS, those early cells. This L1 larval data, it picks up right at that first larval stage, which is pretty common across the Caenorhabditis genus. It maps back to the embryo lineage, sure, but it's describing the cells in the young worm. And the L1 stage itself is interesting, too. It has this L1 arrest state. It's like a pause button the worm hits if it's starting. Pauses development, extends its life. That plasticity is, well, it's a cool biological nugget right there.
Okay, so we've got this incredibly rich, high-resolution data, capturing who the cells are, where they are, when.
Right, all those coordinates
Uh,
and identities.
okay, here's where it gets really interesting for me. How do we go from those snapshots, those lists of coordinates,
Yeah.
to understanding the actual system, the complexity of development? How do we start building models, networks.
That's the critical next step, isn't it? Moving from just raw data to uh, meaningful representations, that deterministic lineage tree we talked about. That's a fundamental one. It's like a structural map of development, the history of every cell. And there are interactive versions online now where you can literally trace any cell back to the start.
Okay, so lineage tree equals family tree.
Yeah.
Got it. But biology isn't just about history, it's about interactions, right? How things actually work together.
Exactly. And that's where network concepts really shine. We can represent different kinds of relationships using graphs. Simple spatial graphs just show which cells are neighbors. Easy enough. But the sources get into the power of hypergraphs. These are graphs where an edge can connect more than just two nodes.
Hold on, more than two nodes on one edge. How does that work in biology?
Think about maybe a process where three proteins have to bind together to function. Or maybe a group of cells that all contribute to forming a single structure. A hypergraph can capture that multi-way relationship with a single hyperedge. The sources even suggest modeling developmental stages as these evolving hypergraphs changing over time. You can add weights to the hyperedges, showing interaction strength, or use dual hypergraphs, which kind of flips the view, makes the interactions themselves the nodes.
And you can analyze the structure of these hypernetworks.
Oh, yeah. You use concepts from graph theory, but extended. Measures based on things like the Laplacian tensor or algebraic connectivity. They tell you about the overall structure, how robust the network is, and this idea of hedge connectivity and hedge cuts sounds particularly cool. It's about finding groups of these hyperedges that, if you took them out together, would kind of break the network apart. Helps you find maybe the critical connections or functional modules within the system. That seems like a real nugget, finding the network's weak spots.
So you can see how different parts are connected, where the really crucial links are. And you mentioned layers. Can you layer these networks?
Absolutely. Biological systems are inherently multi-layered, right? You've got spatial relationships, lineage history, how similar cells are transcriptionally, protein interactions. Multi-layer networks let us represent all these different types of connections at the same time. And there's this concept, node-layer duality, that lets you actually compare distances and relationships across these different layers simultaneously.
Okay, so C. elegans. Its neural network, the connectome, is famous. It's all mapped out. How does that fit in?
The connectome, yeah, the map of all the neuron connections is another perfect example. And like the lineage, it's largely deterministic in the adult worm, mostly the same connections in every worm. But here's a key insight from the sources. There's actually a lot of plasticity, a lot of change happening in the larval worm. Neurons are pruning synapses, maintaining others. It's dynamic. And researchers use these statistical generative models, often based on surprisingly simple things like cell type or when the cell was born, to predict which synapses form and what network motifs appear. The nugget there is maybe that you can predict a lot of this complex wiring using relatively few basic biological rules, but you have to factor in developmental time. Oh, and they even mention work on a glial connectome, the network of support cells.
And beyond neurons, there are all the protein interactions, the interactome, right?
Right. The interactome, protein interaction networks. Nodes are proteins, edges are physical interactions between them. These maps get built using high throughput methods, like yeast-2 hybrid screens. The sources mentioned the WI5 map from way back in 2004 listed something like 5,500 interactions. And a really fascinating biological nugget from studying these interactomes is that when new cellular functions evolve, they often don't build everything from scratch. Instead, they tend to combine brand new proteins with really ancient, pre-existing protein parts. It really supports that idea of evolution as, a tinkerer, reusing and repurposing what's already there.
Okay, wow. So we have the data.
Yeah.
We have ways to represent it, like lineage trees, graphs, these hypergraphs, layered networks, showing all sorts of connections, spatial, historical, molecular.
Exactly, building up the structural and relational maps of the system.
So what does this actually mean when things start happening? How does this structure do anything? How do cells move, tissues fold? How does form emerge? This gets us into the physics of development, right?
It absolutely does, and it's a huge growing area. Because biological tissues, they aren't simple solids or liquids like in a textbook. The sources really emphasize they behave as soft, active materials. They have weird properties like imperfect elasticity, kind of a glassiness in the cytoskeleton. And epithelial sheets, those layers of cells can undergo unjamming, which is like a physical phase transition, lets them flow like a fluid almost.
Biological stuff has its own weird physics.
It really does seem that way. Another idea mentioned is biotensegrity, suggesting tissues might be structured kind of like those tensegrity sculptures, where integrity comes from balanced tension and compression. Modeling that means thinking about forces, movement. And current software, well, it might require some creative workarounds, like maybe modeling biological bits as if they were made of non-biomaterials with known stress strain curves. It shows we're still figuring out how to model these unique materials properly.
And inside the cells themselves, driving a lot of the movement and shape changes.
That's where actin-myosin networks are key players. These are dynamic networks. Actin filaments powered by myosin motors. Super important for cell shape, movement, division. Their function depends on everything the network layout, the mechanics within it, linear, nonlinear, force production. And the biochemical organization is critical too, for holding it together and making force. A key insight here is that you can study these networks even in simplified, minimal systems and learn basic principles. And they show dynamic compartmentalization, assembling and disassembling where and when needed. People use mechanochemical models to try and capture that feedback loop, how forces influence chemistry and chemistry influences forces.
Applying that to a whole developing embryo, that must be incredibly complex.
Oh, it is. Morphogenesis, the whole process of shaping tissues and organs, it's inherently multi-scale, things happening at tiny scales and over long times. It requires collective cell behavior, like cells moving together to fold a tissue inwards called invagination. So instead of just tracking individual cell landmarks, researchers are increasingly using continuum descriptions. They basically turn the cell positions into a mesh, like in engineering, to analyze things like strain rate fields, how the tissue is stretching or compressing over time. And they can check these models by tracking things they can see easily, like cell junctions. Spectral decomposition is a math technique mentioned, helps to sort of untangle different physical processes happening at once by breaking down signals. And some embryos, like ascidians apparently, show these large-scale physical symmetries, like fourfold symmetry emerging from all this.
What about the signals, like morphogen gradients that tell cells where they are? Those are physical too, right? Diffusion and all that.
Exactly. Morphogen gradients, concentration gradients of signaling molecules, are like a biological GPS for cells. Now, how individual cells read that gradient, that's subject to molecular noise, little variations in how fast they make or break down molecules. But here's a surprising nugget. Despite that noise at the cell level, the overall patterns that form are remarkably precise, really reproducible between individuals, which is pretty key for reliable development, you'd think. Measuring these gradients accurately over long distances is tough, though. So people use reaction diffusion models to simulate how they form and how that local variability might play out globally.
It's amazing. Thinking about development not just as genetics, but as this physical process, these complex materials, forces. Okay, let's zoom back in maybe to the molecular level again.
Yeah.
Beyond the physics, what other biological mechanisms, what regulations add more layers of complexity?
Well, one interesting constraint mentioned is something called transcriptional budgeting. Especially in rapidly dividing embryos, cells have to churn out enough transcription factor mRNAs really, really fast to control everything. And C. elegans embryos are apparently incredibly efficient. The sources say they reach production rates close to the theoretical maximum speed RNA polymerase can physically chug along DNA. They know the accumulation rates at single-cell resolution, even study mRNA decay rates quantitatively.
Wow. So they're really pushing the molecular machinery to its absolute limit.
Seems like it, yeah, just to keep up with the pace of division. Another layer of complexity is this infotype heterogeneity. That basically means you can find differences in transcription factor expression within a cell type that otherwise looks pretty uniform, physically or functionally. These subtle molecular differences might depend on, say, where the cell is in the cell cycle, its exact location, or just developmental time. This heterogeneity, backed by quantitative data, adds another layer of functional variation.
And alternative splicing, that thing where one gene can make multiple protein versions.
Yes, alternative splicing, hugely important post-transcriptional mechanism, massively boosts protein diversity from a limited gene set. The mRNA gets cut and stitched together in different ways, making different protein isoforms. The sources mention an atlas of this in the C. elegans nervous system, and it's everywhere, patterns across thousands of genes. Functionally critical, too, controls bits of neuron identity, axon guidance, how nerves find their way, synaptogenesis, and it's even linked to diseases, like some cancers. They find these patterns using techniques like sequencing mRNA libraries.
And C. elegans was central to discovering microRNAs, too, wasn't it? That was huge.
Absolutely, a landmark discovery. Started with that Lin-14 pathway in C. elegans, which controls developmental timing, led to a Nobel Prize eventually. MicroRNAs, yeah, they're small, non-coding RNA molecules found all over in animals and plants. They act as post-transcriptional regulators, usually bind to target mRNAs, often in the 3' UTR bit, and basically dial down protein production. Another really powerful, subtle layer of gene control.
Gosh, so many intricate molecular layers working together. Okay, to study all of this, the data, the networks, the physics, the molecules, that must need massive computational power and, well, really sophisticated tools.
It definitely does. The sources really hammer home the need for a whole range of approaches.
So give us a feel for the computational toolbox then.
Well, we're talking about integrative modeling, pulling different data types together, guided by experiments to simulate biological processes. That involves multi-scale algorithms because things happen at different sizes and speeds, and using statistical ensembles to handle the inherent variability. For the physics side, yeah, specialized tools. CAPI for general physics was mentioned, and even things like MOJOCO and TORAX, which apparently use differentiable physics.
Differentiable physics, what does that even mean? It sounds intense.
It's a pretty powerful idea, actually. It means the simulation is set up so you can easily figure out how changing an input affects the output, which is super useful for optimization like finding the best parameters to match real data or to make a simulated physical system do what you want.
And you can even use computation inspired by evolution itself.
Yeah, things like evolutionary computation, techniques like evolutionary model merging or genetic programming. These methods can sometimes stumble upon really creative solutions to problems, which might offer some clues, perhaps, about how real biological evolution generates novelty. Although, obviously in silico, evolution isn't the same as the real thing.
Where does AI and machine learning fit in? Is it revolutionizing this field?
AI and ML are definitely being used, tools for progress, yes. But the sources are also quite realistic about the bottlenecks. Biological systems are just often way less well-characterized than, say, engineered systems. Experiments fail a lot, knowledge is scattered everywhere. So faster progress needs both AI advances and better wet lab techniques to get more and better data, a more holistic approach. But yeah, people are applying AI to cell modeling, working towards that grand challenge of a virtual cell.
Are there specific kinds of models used for certain biological processes?
Sure, gene regulatory network models, or GRNs, are common for describing early development. They're usually set up as nonlinear stochastic dynamical systems. Analyzing them means looking at their steady states, the stable configurations, and how the system jumps between them, representing cell fate changes.
And I saw something about neuronal input-output functions, like in electronics.
That's a really interesting idea mentioned for systems like C. elegans. With tools like optogenetics controlling neurons with light, you can start to actually measure how a neuron responds to inputs, its input-output function. The analogy is to electrical engineering using transistor I/O functions to model circuits. The hope is maybe you could use these neuronal I/O functions to simulate the whole range of brain states and behaviors.
And are there just, like, practical software tools for visualizing all this crazy complex data?
Oh, absolutely. Software like foam embryo, used with something called polycope, got mentioned, helps visualize the 3D shape of an embryo from segmentation masks, from the imaging data. And the field's mature enough now that there are actually dedicated courses teaching multi-scale modeling techniques.
And that mathematical tool, optimal transport, it kept popping up in the theoretical bits.
Yeah, optimal transport. It's highlighted as a really powerful mathematical framework. Basically, it gives you a rigorous way to think about distances and transformations, but in the space of probability distributions, which makes it super useful for abstracting complex biological processes, which often involve moving or changing distributions of cells or molecules into a mathematical form you can actually work with.
Wow. Okay. We've really gone from tracking single cells, building intricate networks, the weird physics of squishy tissues, detailing the molecular machinery, and surveying these incredibly powerful computational tools, trying to make sense of it all. It's really clear this isn't just one field, is it? It's like a convergence zone.
It truly is. To understand something as fundamental as how an organism develops, you have to bring together insights from physics, computer science, engineering, math, and biology itself. There's no other way.
Let's just step back for a final moment. Thinking about the bigger picture these sources seem to paint, beyond the specific mechanisms, what does this whole approach maybe suggest about, well, the nature of development, of life itself?
Well, one idea that keeps kind of bubbling up is why biology seems so messy. Compared to our engineered things, it's often described as being poised right near the edge of chaos.
Hmm.
But maybe that's not a flaw. Biological systems need to be incredibly plastic, right? Able to remodel, respond to unexpected things, adapt. So that inherent messiness, maybe that's actually the source of its robustness, its flexibility. Maybe it's a feature, not a bug.
That really pushes against the common metaphor, doesn't it? The genome as just a blueprint or like a computer program.
It absolutely does. These sources, taken together, they really reinforce that development isn't just about executing instructions from a blueprint. It emerges from these incredibly complex, indirect, dynamic interactions. Regulation often isn't top-down command. It kind of emerges from how the components interact. It's a much more constructive, interactive process than just running a pre-written program.
And thinking about origins, did the sources mention looking comparatively, like its simpler relatives?
Yeah, there was a mention of looking at the unicellular relatives of animals. Studying how those simpler organisms work might give us clues about how embryonic development, as we know it in animals, could have even gotten started. It also raises the tricky question of how you even define development rigorously. Is it just any sequence of changes? Does it have to be generative, non-degenerate? It gets philosophical fast.
And even with all these amazing tools and insights, there are still major bottlenecks holding things back.
We touched on it with AI, but yeah, it's worth saying again, progress is still often hampered simply because biological systems are, compared to engineered ones, still poorly characterized. We just don't have the complete parts list or a perfect understanding of every single interaction. Closing that knowledge gap is still a huge challenge.
There was a quick mention, differentiation spreading like a wave.
Yeah, just a brief note on that idea. That cells committing to their fates differentiating might actually propagate across a tissue like a wave, and that biomechanochemical models combining the physics and the chemistry might be good tools to explore if that actually happens.
And some of those really intriguing little side notes, ribosomal operating systems, single-cell psychophysics in bacteria. What was that about?
Yeah, just glimpses into how deep the rabbit hole goes. The idea of ribosomes having an operating system suggests incredibly complex internal regulation we're only beginning to appreciate, and studying psychophysics and chemotaxis in something as simple as E. coli. It shows even single cells can exhibit really sophisticated behaviors, like estimation, memory, things we usually think require brains. It just highlights how complex behaviors can emerge from relatively simple rules and codes.
Wow. Okay. We have really covered a huge amount of ground in this deep dive, from the exact coordinates of single cells moving over time, to the complex networks they build, the strange physics governing tissues, the incredibly fast molecular machinery inside, and all the cutting-edge computational tools needed just to start studying it.
Yeah, pulling all these different threads together like your sources do, it definitely paints a much richer, more connected picture. It really showcases the incredible complexity, but also the fascinating problems researchers are tackling to understand how life, well, builds itself.
And as you listening process all of this, maybe consider this final thought. How much of the stunning precision, the order we see in biological development, how much of that might actually emerge not from some rigid top-down master plan, but from dynamic interactions, from inherent variability, maybe even from noise,
Yeah.
all operating right there at the edge of chaos?